Convolutional neural networks are state-of-the-art for medical image segmentation, but creating a sufficient amount of 3D training data can be time-consuming and tedious. We implement a paradigm in which the machine starts making predictions as soon as a few sparse labels become available, and the results can then be refined interactively. Since training CNNs is too costly to achieve this degree of interactivity, we develop a hybrid architecture in which an unsupervised dual-branch CNN pre-computes features that have been specifically optimized for use with a fast random forest classifier. We apply this framework to visualize multi-shell diffusion MRI, an intricate high-dimensional type of volumetric data for which few visualization options are currently available. For more details, please watch our presentation and read our paper.
How often have you relied on the histogram when exploring a new data set? But what if the histogram actually hides interesting features? You might have heard about alpha histograms where multiple frequency distributions are combined into a single histogram, thereby revealing previously hidden features. This work extends this idea by keeping the local distributions around and using them as input for a similarity-based hierarchical clustering. As the clusters refer to features within the dataset, we can establish a link between attribute space and the physical domain of the data. This enables the exploration of features by either selecting cluster representatives or navigating through the hierarchy.
In this project, we are developing a cost-effective virtual reality solution for training endoscopy procedures. An endoscope is a fiber optic bundle with a camera at one end which can be inserted into a patient. The picture on the left is showing an endoscope controller. To know the amount of insertion or withdrawal of the endoscope, we are tracking a rod in our virtual environment, as shown in the video. The tip of the endoscope can be reorientated by using the dials on the controller, simulated here using a Microsoft dial device. When wearing the head-mounted display, you see a patient and there's a monitor showing the endoscope view. The video on the right is replicating that. There's a polyp. You'll see that we can change the orientation of the camera. We can even simulate insertion of a CO2 gas into the anatomy in order to inflate it. More detail in our presentation.
Virtual reality platform for training and planning implantations of occluded devices in left atrial appendages. The left atrial appendage occlusion is a surgical process that involves implantation of an occluded device in the left atrial appendage. The VRITA platform allows to plan the implantation of an occluded device with the help of other platform functionalities, as for example the visualization and navigation through CT images, the visualization of left atrial appendage morphological measurements, the change of transparency of the left atrial mesh, or the clipping of the mesh in order to better visualize its 3D morphological aspects. Regarding the implantation, the user can move the device through the left atrial center line in order to change its position. The implantation can also be done with a transeptal catheter model that works with an endoscopic mode. Concerning the results, VRITA has shown to better visualize the leaks between the device and the atrial wall. Finally, the platform has been tested by 5 users before answering a questionnaire, where VRITA has been positively rated as a training tool and as a preparative tool, but with some improvements to be done in the ease of use.
In time series experiments studying gene expression, we can compare samples to a reference to produce lists of differentially expressed genes. However, these lists are very long and hard to interpret, so we need geoterms to come to the rescue. With geoterms, we can categorize the genes like we're finding all genes associated with translation in this list. For each of our lists, we look for terms that are significantly overrepresented in comparison to the background, resulting in lists of geoterms with associated p-values. These lists are still quite long. Can we make them even shorter? Can we compare them visually? Yes, with our method. Some geoterms are more dispensable than others. We cluster terms hierarchically based on their dispensability and filter out redundant terms using a slider. We produce flat clusters by cutting the tree. Each clustered list is represented by a tree map, and we can compare them, for example, using animation. Please visit us at our poster.
Sequence diagrams are a common way to visualize the primary structure of a protein, that is, its amino acid sequence and other attributes. We present an interactive web-based sequence diagram which was implemented using the popular JavaScript library D3. Besides the attributes provided by the Protein Data Bank, we can also visualize derived data provided by external tools. We extended the idea of traditional sequence diagrams by offering the option to encode suitable attributes per atom instead of just per amino acid. Our interactive sequence diagram includes tooltips and is fully configurable. For example, it is possible to switch to line charts or bar charts to represent certain attributes. Our tool is still work in progress. In the future, we want to link it to a 3D view and to use it for the visualization of multi-sequence alignments. If you're interested, we would be glad to talk to you about our tool and discuss more ideas for possible extensions. Please join us in the Discord channel of our poster.
I would like to present Graph Layout by Path-Guided Stochastic Gradient Descent by Simon Hoymus, Andrea Guaracino, and Eric Garrison. Pangenome graphs built from raw set of alignments may have complex structures which can introduce difficulty in downstream analyses. Our proposed algorithm aims to find the best node order for a 1D and 2D layout in order to simplify these complex regions. Our idea is to move a single pair of nodes at a time, optimizing the disparity between the layout distance of a node pair and the actual nucleotide distance of a path traversing these nodes. As an example, on the left is an unsorted Pangenome graph in 1D. The presence of many and long links highlights that the node order is not optimal. Applying our 1D path-guided SGD, we obtain the sorted graph on the top right. The animation below shows how the algorithm works. Our algorithm can also be applied in 2D.
Molecular representations are taking on an important role in communicating ideas and generating new hypotheses for biological mechanisms, and in analyzing molecular simulations. Virtual reality headsets offer performant and accessible solutions. However, software adaptations are necessary if we wish to fully benefit from virtual reality in a scientific visualization context. This poster presents a few examples implemented in UnityMol, our molecular viewer and visualization platform. We present two scenarios where VR has a significant edge over standard desktop setups. Molecular exploration greatly benefits from 6 degrees of freedom interaction thanks to VR controllers. Interactive molecular dynamics simulation sessions are vastly improved using rich interaction devices and depth perception.
We present a medical application for the visual analysis of multivariate, time-varying patient data from intensive care units. In order to visualize this multitude of measured variables in a clear, meaningful way, we employ dimensionality reduction to project the multivariate intensive care data to a two-dimensional space. We generate a time curve using these projected data points. This curve is a connected path indicating the progression of the patient over time in the high-dimensional space. That is, path segments that are close to each other indicate a similar patient state. The time curve provides an overview of the data and is linked to additional plots for in-depth data exploration. To utilize the concept of time curves in a multi-patient scenario, we also use data depth to find and visualize representative patients and outliers in an ensemble. We showcase the feasibility of our approach on data collected from a set of pigs who were monitored in an intensive care unit during a controlled experiment. The aim was to gain insights about patient state changes with respect to a varying fluid balance.
In our study, we take you through the iterative design process that led to GLANCE, an application for monitoring glaucoma progression using deep learning in the clinic. Deep learning is increasingly used in the field of glaucoma research. However, issues with trust, interpretability, and practical utility form barriers to clinical adoption. We explored how visualization can be used to overcome these issues. The current tool is the result of an iterative participatory design process together with clinicians and manufacturers of OCT machines. Together, we identified tasks and design goals, because clinicians have different needs than researchers and developers. Us researchers love to explore, but clinicians need to make immediate diagnosis, and therefore want fast and easily retrievable results. We show historical behavior of the model and combine clinical information with activation maps in a way that is familiar to the clinician. Our approach demonstrates how visualizations of deep learning models can augment clinicians' knowledge and calibrate their trust during clinical decision-making.
There are plenty of decision support systems used in everyday life. Trip planners, for example, assist users in finding the best route to a specific destination while providing additional information, such as traffic jam on the way, the approximate duration for the ride, and possible alternative routes. With these information entities in mind, users gain trust into the recommendation and follow the advice. In clinical decision support systems, however, these additional information entities needed to gain trust into the system are not well studied, and up to date, no perfect explainable clinical decision support system has been developed. We are addressing this challenge by identifying the required information, allowing physicians to comprehend the recommendation process, and developed a prototype for this purpose. Come and listen to my talk about a visual approach to explainable computerized clinical decision support to hear more about our work.
Comparing proteins among each other, especially visually, is no easy task. It may be even hard when the exact same molecule is shown. We therefore created an approach that utilizes the so-called molecular surface maps. Using several metrics for the determination of the distance between images, we are able to cluster the proteins visually. Our approach is able to take multiple surface properties into account to create the clustering. We finally compare the results to already existing similarity metrics, including a database for functional similarity. It is found that our approach generates good results, although the amount of information used to retrieve them is limited compared to other methods.
Hello, we present a 3D visualization of amyloid beta fibrils from 2D cryo-EM density maps. During the last years, resolutions of cryo-EM density maps were growing significantly, getting closer and closer to atomic resolutions. With the growth of resolutions, also dataset sizes become larger. Further, during research, a direct side-by-side comparison of multiple fibrils is needed, also increasing dataset sizes. Hence, we propose an adapted ray margin algorithm to visualize long sections of the fibril without requiring additional GPU memory. In our results, we can show that our approach is not only able to outperform standard volume rendering regarding memory consumption, but also regarding performance of large volume renderings. Lastly, we implement state-of-the-art illumination techniques and propose a simple yet effective visualization of data uncertainty, which is introduced by the previous reconstruction process. We hope to see you at the talk.
In structural biology, the scientists study protein flexibility by decomposing protein movement into normal modes. Each normal mode describes a particular oscillatory motion pattern of individual atoms. The major challenge is that normal mode calculation yields a large number of modes, typically 3 times the number of atoms. Therefore, we present a new interactive and visual approach consisting of several linked 2D and 3D views to aid with the analysis of this high-dimensional normal mode space. The 2D views display the influence of individual normal modes or their combinations on the protein voids, thus enabling localized analysis. The 3D view then provides various representations, for instance glyphs, coloring, or different rendering styles, to study the normal modes and their influence on the protein globally. Finally, after the user identifies the possible functional movements, he or she can visualize the realistic movement of the protein driven by these modes.
Cerebral aneurysms are pathological dilations of cerebral arteries. They bear the risk of rupture, which is associated with a high death rate. We developed various methods to support risk analysis and therapy planning of cerebral aneurysms. First, we developed an automatic calculation of morphological parameters. We also developed techniques that allow simultaneous exploration of multiple scalar fields on the vessel wall to identify high-risk regions. In addition, we explored techniques for simultaneous analysis of wall and flow properties to better understand the interaction. In addition, we designed a comparative representation of tensor data representing mechanical stresses in the vessel wall. To assess blood flow complexity, we developed a method to first cluster integral lines representing blood flow and subsequently classify the resulting flow patterns according to predefined types. All methods were integrated as modules into our developed system called Analysis. The user is guided through the exploration of the complex simulation data to assess the rupture risk and to find an optimal treatment.
Measurements and simulations are widely used to generate multi-field ensembles of tubular flow. Combining both domains might eliminate the respective drawbacks, such as low resolution or choosing good simulation parameters. We use our novel similarity measure to create a low-dimensional embedding for such ensembles. Similarity between measurements and simulation runs can be assessed by our overview visualization. Below, we see the spatial similarity distribution for selected runs, as well as streamline visualizations. Selecting the measurement and the most similar simulation run, we can inspect the actual difference by placing an arbitrary plane from which we derive a slice-based representation. See you at our talk on Thursday at the Vascular and Flow session.
Welcome, this is Virtual DSA++, an algorithm for the automated segmentation, vessel labeling, occlusion detection and graph search on CTA and geography data. Virtual DSA++ comes with a lot of features. First, it leads to a fully automated segmentation and modeling of the cerebrovascular vessel tree, and only a CT dataset is required. It also allows to exclude veins from the rendering based on their shortest distance to the circle of Willis. This feature is called vein suppression. The blue cube in the center indicates the root node where the vein suppression relates to. Virtual DSA++ also allows to interactively search for shortest pathways just by clicking into the desired point. The feature is meant to be used to plan mechanical thrombectomies, for instance. It also automatically labels the cerebral arteries medial left and right, posterior left and right, and anterior. Last but not least, it also can detect occlusions in the named vessel. This might be useful for physicians in the diagnosis of ischemic strokes. Thank you for the attention.